In bacteria, there are strains of bacteria that can recognize self versus non-self, and they'll form borders. Social amoeba, like Dictyostelium, also segregate according to their genotype. Fungi have two completely different systems for mate recognition and preventing somatic cell fusion. Flowering plants have three systems that prevent them from selfing. Sea squirts have self-incompatibility systems, and of course, colonial invertebrates have these systems. In each of these cases, evolution has taken a problem, which is how do you distinguish between members of your own species? And it appears to have invented new and different ways of doing that. So I think it's a really interesting example at a molecular level of the evolution of novelties. And in particular, I think the hydroactinium system is novel in a way that I've been talking about this whole talk, which is that if you look at how these systems are built in slime molds, in flowering plants, and in fungi, they're usually two tightly linked genes, where one is a receptor and one is a ligand. And so variation in the receptor and the ligand has built up over time, but there's a lock and a key, and there's always tightly linked, so that preserves the integrity of the response, right? Or the integrity of the identity. In hydroactinia, there isn't a lock and key, but it's this one molecule that's binding only to itself. And how a system like this evolves in comparison to how a system like these evolved at a molecular level is, I think, something that will be interesting to study as we go forward. So with that, I'll just say that the work that I showed you here has been done mostly by Uma Karaj, who was a former postdoc in the lab. Steve Sanders and Aidan Hune are a postdoc and grad student in my lab. And Drake is our lab manager and makes sure that the animals are all happy and reproducing and lets us do our work. So I have funding from a couple of different sources. And in particular, we have just a great group of collaborators. It's a very small, as you might imagine, it's a small field. But I'm really lucky in that all the labs that are working on hydroactinia, we get along very well, and we've come together for this genome project, which has been really fantastic. So with that, I will stop, and thank you for your attention and take any questions.